That's very appreciated. Okay, so you should have been able to find yesterday's lecture online. Just as we did yesterday, today's description on YouTube will have the link directly to the slides, but you already have it in this class as part of the Google folder. So I'm going to start us off with our title slide. So today, we're moving out of the whole computer science realm and starting to talk about Turkey. We're going to discuss the problem of genetic variation and aligning that with phenotype, understanding how the differences we see genomically map to phenotype. So we're going to talk about DNA sequencing and variance detection, and I hope that we'll be able to cover quite a few things. Now this is sort of a partial overview of the topics we'll address, but the first of these relates to Phred scores. This is something that I've mentioned in several contexts. In this case, how do we assess the errors associated with base calling when we're doing sequencing? We will talk a little bit about Bowtie, but maybe not as much as it would suggest by having it in this overview. Bowtie uses an awful lot of special tricks to align read sequences to an annotation. And then finally, we'll talk about Genome-Wide Association Studies and several things pertaining to that. Topics like stratification, admixture, linkage, and Manhattan plots, an old favorite of mine. So let's plow ahead. Now, I've mentioned before that this diagram gives us kind of a roadmap of where we sit. Today's the second day in our story, so you can see that we're going to come right down the center line in the genomics slide. So we'll be talking about the problems of base calling, of read mapping, variant detection, admixture analysis, which is something that has relevance in the United States and certainly here as well, and finally Genome-Wide Association Studies. Let us start with an electropherogram. I feel that it's important that everybody know what an electropherogram is because they are the essential bit of data from which, not technical bits, but they're the essential source of data from which we derive sequences. So that this problem of base calling algorithms, is one of working from an electropherogram to produce sequence, raw sequence information. So when we look at this electropherogram, we're seeing 4 different dyes rising and falling in intensity as a function of time. This is from an old capillary electrophoresis instrument. As I mentioned, capillary electrophoresis instruments were capable of giving us on the order of 500 or 600 nucleotides of sequence for an individual read. So that's pretty great. And you can see why as you look at this. You can see that, say around 300, we have this tiny little pip at 300 in blue. That represents a C. Here we have 3 little peaks ahead of that are in red. Those tell us that those are T's. Then we have a whole series of A, each marked by a well-resolved rise and fall in the A channel. So that's grand. We're measuring 4 different intensities for 4 different dyes, 4 different intensities associated each with a different dye, as these products of polymerase are flushed off of the capillary electrophoresis column. So that's grand. Now, if you look at the sequences, or at the series of the electropherogram around 300 or 200, you're kind of looking in the sweet spot of these runs. You have a nice rise and fall, and when the signal in fluorescence falls, it falls all the way to the baseline, so that we have completely resolved neighboring peaks for neighboring amino acids. But look what happens once you get up here into the 700s or even 500, 600s. Look at this little region of 4A nucleotides around 680. So you have this sort of waves back and forth a little vaguely across those 4 bumps. How sure are we that these 4 A's are not 3 A's that just have some noise added to them? That's kind of a problematic thing. So as we frequently see in all kinds of separation, the smaller the fractional difference you're trying to separate, the harder it is to accomplish for some separation device, even capillary electrophoresis, which is quite good at separation. So by the time you get to this smear out at, say, 736 or so, we have a whole bunch of A's, but we're not really sure how many of them there are, because there's barely even a wiggle up and down in that mound to tell us how many A's they are. So how, if we were trying to be very simple about this. We might say there's a first nucleotide base that we feel comfortable about, and there's a last one, and anything outside that we're going to just throw out. Okay? That's a really basic kind of rule. We'd say anything past, I declare, 600 shall be trash, and anything before, let us say, 50 shall be trash, and we'll accept everything else. That's a discretization. It's separating these reads into the good and the bad, and everything else, and that's it, right? So it's a very simple-minded way to go. Some researchers at the University of Washington realized that they didn't want to go this route, that instead they wanted to make their claims about accuracy on individual base calls. So not trying to figure out which part of the sequence we're going to accept, but rather saying, what is our probability of error associated with each letter in the sequence? That's a lot harder, and some of this was motivated by the fact that, as I said, the legislation that enabled the Human Genome Project had a specified error rate we wanted to accomplish. So the problem for Phred is how can we estimate base calling errors? How can we estimate the probability that an individual letter is an error? So we're going to start by having to infer a sequence. We're going to have to start at some point by saying, what letters are we assessing the probabilities for? From there, we're going to have to associate a probability with it. So there are actually a pair of papers back-to-back in genome research from 1998 on this subject. These came out, just for context, I always find it's useful to sort of set these against the scope of history, and what I have is my own life. So I was at the University of Washington, having become a grad student in 1996, and 2 years after I was there, these papers were published on how to compute Phred scores. Now, you might ask, why do we keep calling them Phred scores if we're no longer using Phred for computing them? After all, we're in the realm of high-throughput sequencers now, massively parallel sequencers. So why are we still calling them Phred scores? And the answer is that the way that Phred computes its scores has been adapted to apply to the newest sequencers, and the way it expresses errors is a very good universal model, really, for talking about error. So how do we separate a good base call from a bad one? Now, the paper is fairly complex, but I'm going to try to boil it down as much as I can. First off, peaks that we see in the electropherogram, these rises and falls, are going to match with the beat, boom, 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 of the electropherogram. So we're going to need to look for a rise and fall at regularly spaced times in this electropherogram and see that we have a good peak that matches with that rhythm. Only one of the traces is concave down at this call. Now, how many people here have had calculus? A little bit of calculus, maybe some in college? A few of you have, okay, that's good. I was advised by my undergraduate advisor at the University of Arkansas, if I wanted to go into bioinformatics, that I should probably take at least a year of calculus, and so I did that. And I gotta say, it was painful. It was painful. Calculus never flowed for me the way that like analytical geometry did, but it does have some value. And I want to point out that concave down should have a special meaning for you if you've been through calculus. Because when we think about the first derivative of a thing, we get its slope, as some of this coming back to people. And if we take the second derivative of it, we figure out if it's kind of a u up or a u down. Oh, great. So now that we've all had that little shock where we had to remember a little bit of calculus, let's just think that we want something that's pointing down, right? We want we want an inverted valley. Similarly, there are 4 different traces at each time point, and we're arguing that only one of them is going to be concave down here. So it's not fair to have both a C and a G going up and down together at this location. It's no good. That means that we've got some sort of disagreement within our data about what base call is there. So we want only one of these 4 tracks to be a nice upside down U. Next, when I first moved to Nashville, Tennessee, I moved into a neighborhood called 12 South. And when I moved there, 12 South was kind of evolving in how it had, the role it had played in Nashville. Because it used to be that my corner at 9th Avenue and Montrose Avenue was the great place to go if you were looking for crack. So when I moved there, some people had bought the property, hollowed it out, and basically rebuilt a building on the scaffold of its guts. And so it was a very nice house when I moved into it. And yet, only a couple of years before I lived there, you could find needles on the property. Weird. So, imagine that you're in a neighborhood that has a house selling drugs. If you are the next door neighbor to the house selling drugs, your property values are going to take a bigger hit than if you're 5 houses away, or half a mile away. Many kilometers away is even better, right? So imagine that if you have a really lousy looking base call somewhere, if you're right next door to that, your score is going to be compromised because you're close to the house selling drugs, okay? If you're further away from that, you can place greater confidence in your accuracy. Okay, so those are 3 rules that we can use to put some sort of error status on an individual base call. But I'm going to try to dig into these just a little bit. The first of these comes because Fourier Transform is used everywhere in bioinformatics. And I think it's really important that everybody has some kind of intuition about what a Fourier Transform is. No judgments, but some of you have been to a party at your friend's house who's got like the really oversized stereo, right? And some of us even danced a little bit. Now my dancing is not worth capturing on camera, trust me on this one. But I want you to think about the particular qualities of house music. What is the signifying characteristic of house music? What I'm looking for is something like lump, 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 lump. It's a strong and firm and persistent beat that you can hear throughout. So I want you to think of this whomp, whomp, whomp as producing a very low frequency that can be found in the sound. So this is a change in amplitude with time that our ears experience as a low frequency coming at these spaced out. In Fourier Transform space, maybe on your graphic equalizer if you've got one of these, you can see these low frequencies by a bar that's jumping up and down in tune in beat with the music at the low end of the graphic equalizer. That's because it's showing a Fourier transform of the sound you're hearing. It's decomposing the sound that you hear into its component frequencies and visualizing that. So a Fourier transform, wherever we see it throughout bioinformatics, is generally playing the role of detecting frequencies. Okay, so if we imagine, if we take a look at our diagram on screen, oh, here's my laser, there we go. So we start with this function of time. Here we see that over time we have a rise and a fall in the signal amplitude. It has some characteristic frequency that we associate with it. Now this is a relatively low frequency compared to this. Do we agree? So this rises and falls about half as quickly as does this. So when we look at it on the frequency domain, we get a positive and a negative part of this equally spaced out from 0. That is strong, indicating that this top signal is rising to a higher maximum and a lower minimum than this one, but further away from 0, indicating that it's a longer wavelength or lower frequency, same thing. Here we see that we have a smaller amplitude, thus smaller peaks over here, but it's a faster frequency, which means a higher frequency, which means it's further from 0. Okay, so far so good. We have a pattern of intensity with time. We've mapped that to a particular frequency. These are sine waves, right? They have just frequency and amplitude, essentially. So let us now imagine that we have two signals that represent an addition of these two. So we have the slow moving up and down in greater magnitude here, and we have a fast moving up and down with a smaller travel on it. When you add those together, you get this complex waveform. When you look at it in Fourier transform space, though, it's a whole lot simpler to analyze. This plus this equals this in frequency space. This plus this equals that in time and amplitude. So Fourier transform is our ticket to transforming complex signals into a set of frequencies that have been added together. So I mentioned just a moment ago that we want to be able to match peaks to the characteristic frequency of the electropherogram. The electropherogram being this pattern of fluorescent intensity with time. So I want you to imagine that we have a sort of problematic space in our electropherogram. Now, at this point, I'm not separating out colors one to each of the dyes that we're using. But I have a set of predicted locations and a predicted period for this as well. This is to say that Fourier Transform has looked at the overall signal and feels that we have hits on where the peaks should appear at these regularly spaced locations. And it knows how big a distance it expects between successive locations. So from FT, we have these predicted 4 locations. These, however, are the data that we have. So if I'm trying to make an assessment of a particular peak, I can ask, how far is its centroid, or its middle value, its maximum value, from the closest predicted beat of the electropherogram? If those are far apart, we're a little worried about that, but we're most concerned about it as a function of how much this difference between predicted and observed peak times are compared to the overall predicted period. Okay, so that is one of the ways that we can use Fourier Transform to infer a frequency out of the electropherogram and ask whether an individual peak matches with that overall pattern of peaks and valleys. Everyone good with that? Okay. That is not to say that Fourier Transform is only involved with Phred, because it shows up anywhere you have a bunch of frequencies in mass spectrometry, for example, but in lots of different fields. Okay. The other thing that I think Phil Green did really, really well with formulating Phred was how it expresses its scores. And this, more than anything else, represents Phred's major contribution in bioinformatics that continues through, and why we still call them Phred scores, even if we're not using the Phred software to do them. This is to say that we start with some probability. This is a probability of error, not a probability of correctness, so it's kind of turned inside out in that respect. So if you have a very, very low probability of error, that's important to know. And we want to accentuate the difference between a 1 in 1,000 probability of error, a 1 in 100,000 probability of error, a 1 in 100 probability of error. We want to be able to really tell the difference between these very small numbers. One of the best ways to do that, he found, was to use the negative log of that number. Does everyone remember logarithms and exponents? That's kind of a challenge for us. So let's start with how this works out. You might start with a 1% probability of error. That's to say 99 times out of 100, our call on this is right, but it could be erroneous just the same. So at a probability of 0.01 can be written in scientific notation as 1 times, well, 1e minus 2, which is the same thing as saying 1 times 10 to the negative second power. Does everyone remember what 10 to the negative second power equals? 10 to the negative second power. Let's start this way. What is 10 to the first power? 10. Great. Okay, that's good. What is 10 to the 0th power? 1. It's 10-fold less. What is 10 to the negative first power? What was that? Okay, we sort of fell apart there. So 10 to the first power is 10. 10 to the 0th power is 1. 10 to the negative first power is 1 over 10. Tricky, huh? So we never get down to 0, but we're getting infinitesimally close to it. So let's just work through the series for a minute. 10 to the first is 10. 10 to the 0th is 1. 10 to the negative first is 1 over 10. 10 to the negative second is 1 over 100. 10 to the negative third is 1 over 1,000. You see where I'm going with this? These are the kinds of probabilities we want to accentuate. So when we have our very low probabilities, we can convert that to scientific notation. We see that this is 1%. But you see, this little number right here is what we're actually going to care about. Because the more negative this is, the more positive we want our score to be. So the formulation they chose is negative 10 times the log base 10 of p. And a shorter way to say that is that we're going to grab these exponents, like negative 2, make them positive, and multiply by 10. So this minus 2 becomes 20. This minus 3 becomes 30. This minus 4 becomes 40. And so when we look at these numbers, a Q value of 20 it indicates a probability of error for this individual base call of 1 in 100. Some people ask if I ever ask math questions on my quizzes. I do sometimes, most frequently about logs, because I find that that's something that everyone needs to know really, really well. I'm not saying I did, I'm just saying that this is the sort of thing I ask about. So when we see a 20, I want you to read that and immediately say, that's a 1% chance of error. When you see a 30 for Phred, I want you to think that's a 0.1% chance of error, or a 1 in 1,000 error. When you see 40, I want you to think that's 1 in 10,000 chance of error. Okay? So Phred scores, the score formulation, is a way to highlight how close to 0 this probability of error really is. Okay, on we go. Now, we mentioned FASTQC this morning, but now that we know what we know about Q values, the Phred scores for individual base calls, I want you to think again about this graph that we saw. Now, this is a different graph than we saw. This morning, I was working from some data I worked with from Chia. This is another, just a sample diagram provided by the folks at B. Abraham, I'm not sure, who created this software to begin with. So here we can see then that we're getting a slight diminution as we go to higher and higher base numbers within a read of increasing error. If you think back to our electropherograms just a moment ago, you saw that our ability to separate next door neighbors started vanishing, especially as we got to these really high base numbers. That principle is not really that different from capillary electrophoresis to today's massively parallel sequencers. We still see that our ability to distinguish base calls as we get further and further into the sequence diminishes. And you can see that in the FASTQC report here. We see that the minimum values that we're seeing are starting to sag as we get further and further through the sequence read. Okay, so I realize that there's a lot of jargon surrounding sequencing data, and I wanted to take just a moment to try to work our way through some of those. I've been using the word read a lot. In this case, a read represents an individual sequence produced by the sequencer. You may have one piece of DNA where you have a read on either end. That's going to come into particular emphasis tomorrow. So those would be called paired end reads. So we frequently refer to the DNA that we're trying to sequence, the piece of DNA that we're trying to sequence, as the template. So if I use the word template, I want to make sure you know that. Shotgun sequencing is a really popular technique, and it's really grown in how much people use it as time has passed. In the old days, sequencing something was really expensive and fairly slow, and as a result, we spent a lot of time trying to figure out which pieces of DNA are best for us to sequence next. Whole genome shotgun is a rather different approach. The idea here being that each sequencing reaction is cheap, and we just need to keep the sequencer busy, and soon enough, we'll have enough data to sequence whatever we want. So we've shifted from this world where we would carefully pick which bits of DNA needed to be sequenced to just sequencing everything and hoping bioinformatics will sort it all out. Okay, fold coverage. This is a term that shows up all the time in our publications on this subject. In brief, we want to know, on average, how many bases of read have we generated compared to the number of actual nucleotides in the organism. So in the old days, we would do sequencing of non-model organisms at really low depth. So things like the first sequencing of the American alligator took place at 2x coverage, which is to say that large portions of that genome were not even touched by the sequencer, but quite a few of them had stacks of sequences. But on mean, we had 2 times as much sequence as we in reads as we did in the actual genome itself that we were trying to model. Okay, so high fold coverage means that you're going to have a much better shot of seeing everything and having greater confidence in an individual variant, for example. than you would if you had very low fold coverage. Contig is a term that's going to show up a lot tomorrow when we're talking about assembly. Contigs refer to the fact that these individual reads are not going to remain by themselves for long. We're going to be looking for ways that they overlap. And so contigs are going to be built that stitch together short read sequences to make a longer stretch. Now, in the old days, we used to moan an awful lot about the fact that we just didn't have sequence in some of the places we wanted. You know, maybe you've got your whole chromosome sequence that your chromosome center has been assigned, but you have 4 or 5 pieces and you don't know what falls in between those, maybe because of repetitive DNA. So in the old days, closing used to consume an awful lot of effort on the part of genome sequencing centers. So we're going to talk a little bit more about that stuff tomorrow, but I didn't want those terms to just drift by without our having mentioned them here. So how does a sequencer work? Well, massively parallel sequencing is built around this concept that if we can get individual DNA molecules separated from each other, we can then amplify up a whole bunch of copies of those, typically by something like PCR. If you remember from the video this morning, we looked at something called bridge PCR as part of that YouTube video. So if you weren't able to be here this morning, you can just see the YouTube link in the write-up on the Google Drive. So being able to synthesize many copies of a piece of DNA at some location in the sequencer is necessary. You'll see some people use things like emulsion PCR or bridge PCR, whatever. But think of PCR as our molecular photocopier. We're going to make a whole bunch of copies of some sequence at some location in there. And then we're going to get to see things as kind of a function of time. We're going to get different snapshots of each strand. So I want you to think for a moment about these 5 pieces of, these 5 frames, and I want you to think of them as though they're a cartoon. It may not be obvious at first glance, but each of these, if laid on top of the one before it, will position a dot over this. So think of these as like cells in a cartoon. Our first glimpse of these 4 different sequences that we're trying to sequence shows a T at this top one, a G to the left, a C at the bottom left, and an A to the bottom right. Our next snapshot shows the incorporation of the next nucleotide at those 4 locations. We see where before there was a T, there's now an A. And later on we see there's another A, and then a G, and then a T, for just this top position. In the left position we see that we had a G, then we had a C, then we had a T, then a G, then an A. So in effect, we're looking at the same spots as a function of time, and these flashes of light that we're seeing at these locations tell us about successive addition of nucleotides to a particular sequence. Okay? So, in this case, with these 5 images of 4 locations, we're building out sequences for each of these spots. So when we look at the example at the left, it started as, its first flash was a G, its second was a C, its third was a T, its fourth was a G, its fifth was an A. We infer, for that position at the left, this sequence as a result. So we're going to start with random locations of these DNA molecules. Successively, we're going to add on a complementary strand, and the order of those flashes of light is going to tell us what the sequence is at that location. Okay, now I found that a little hard to get my head wrapped around, and the first few times I saw people speak on how these sequencers work, I found myself mystified. So if it is not making immediate sense to you, there are lots of videos on YouTube about this subject. I'm not actually expert in it, but I'll try to help where I can to fill in that hole. Because we use sequencers for bloody everything these days, and it's awfully useful to know the nature of the data that underlie those sequences. A lot of people assume that any new technology is clearly better than any old technology. And I want to rescue people from that notion. Newer is not always better in all respects. And one of the problems that we encounter is that today's sequencers tend to produce a lot shorter sequences than what we are used to. So I showed you an electropherogram from capillary electrophoresis. I think you can see how you could translate those peaks and valleys into 500 or 600 base pairs. Most sequencers today, however, use short-read sequencing technology. That's not to say this is the only game in town. There are certainly instruments out there that can sequence very, very long strands of DNA, far longer than any capillary electrophoresis device ever could. But in the main, when people are talking about generating megabases and megabases, they're talking about doing so with things like Illumina sequencers that use relatively short reads. So I've written in that in the old days we got 600 base pairs and now we get 150. But that's really quite sloppy, because as we've seen, some of the modern sequencers are generating 50 residue sequences. So, and it's not even the case that the best sequencers are producing the longest reads either. For example, there's a relatively old machine called a MiSeq over at UWC, and the MiSeq produces longer sequences than the very expensive HiSeq X10s that we have in Beijing, knocking out genome after genome. So you're not necessarily going to see a correspondence between length and excellence of instrumentation or price of instrumentation. In this case, I'm using Sanger as a synonym for old school, I guess, in sequencers. So we tend to think of Sanger sequences as producing higher quality scores for individual base calls. That said, it's definitely the case that modern sequencers that produce huge numbers of reads frequently cover the same region many times over. So even if you have a lower average base call score for modern sequencers, because you have so much depth on these locations, you can feel very confident that the effective error at an individual read, at an individual base, is going to be much lower than if you tried to do it only with Sanger sequencing. That's paradoxical. You can have lower quality, but because you have a whole lot of it, you're able to get greater confidence in the individual base calls. Okay, so in the end, the chief advantage that we get from massively parallel sequencers is that they are sequencing an incomparably greater number of templates than any capillary electrophoresis instrument ever did. So our goal, our chief design aims with today's sequencers are about volume, not necessarily about length, not necessarily about the quality scores, but by just covering us in data, they help us to accomplish all of the goals that we have. So why do we need to emphasize the alignment problem for short reads? So now we're making our shift from the technology itself, from the instrumentation, to how we manipulate the data from it. So we have several options. One would be called a de novo assembly. A lot more about that tomorrow. De novo assembly means that we try to just stitch all of these short reads together into longer and longer contigs until we're able to explain all of the data. That is possible, but it's also going to take a lot of computer time, a lot more memory than your laptop has to offer, and it tends to be a bit error prone. So that's not great. We would also care an awful lot about recognizing sequence variants. We would like to know when this person has a variant form of this gene, a variant allele, than what the reference annotation would suggest they have at that location. So aligning gives us the ability to compare a standard reference against what this person displays at a location. We also have this kind of time game. Having even a few massively parallel sequencers around means that you're going to be playing catch-up all the time, trying to accommodate and trying to understand all the data that these instruments can keep. If you are using your instrument one day a week and spend the rest of your time playing catch-up on your computer, that's a really bad scenario, because it means that an expensive instrument is lying idle because you haven't got enough computer people, basically. So if you can instead use alignment, you can have a much faster analysis of the data from the sequencer than if you attempt to do a de novo assembly. And also, it's quite important that we understand that all the downstream stuff we want to do, like doing variant calling to produce variant call files, for example, is going to assume that you've been able to do this alignment already. So having a QC step, having some sort of trimming to get the nonsense away from these sequences, and having an alignment to a reference annotation is just part of the game. It's what you have to do in order to get to the next stage of data analysis. So one of the algorithms I want to point you to is called Bowtie. Bowtie is part of what frequently gets called the Tuxedo suite, and as such, it's a very big player in this game. I want to note that Burrows and Wheeler, the two guys who are behind this algorithm, are not known for Burrows-Wheeler, oddly enough. Burrows created this little thing called a subroutine. And if you've ever had a programming class, at some point you've written a subroutine. Well, the person who named them is Mr. Burrows. Now, Wheeler has another interesting story. Back in the day before everybody used Google to do a search, there were many search engines that were all competing, things like Dogpile and AltaVista and so on and so forth. Some of us may be old enough to remember that time. That's okay. So Wheeler was the person who created the AltaVista search engine. So we see that you don't have to be Sergey Brin to get involved behind Google to be involved in all sorts of other activities. The people behind the AltaVista search engine came to bioinformatics and found interesting problems to solve here. Burrows-Wheeler is a fairly complex bit of algorithm, and I feel that spending a lot of time trying to discuss it is going to be challenging. So if you are really interested in computer science, I would suggest you follow that URL link down at the bottom, and you can learn the story of how Burrows-Wheeler works. The short version, about the only thing that I really want to be sure everyone understands about this, is the concept of a suffix array. So if you imagine that you have some sequence, you can imagine that you could chop off the first letter and you'd get a subsequence of that. It has the same terminus, the same right side, but you've now lopped off the first character of it. And you could lop off the first two characters of it and get yet another suffix sequence from it. So from a long sequence, a sequence of n letters, you can get n-1 different suffixes from it. So a Burrows-Wheeler suffix array uses the structure, remembering what letter came before, in order to do a very rapid search of a sorry, it allows for a bisection search, one of which I mentioned yesterday. Do you remember when we were talking about binary search trees? And I mentioned that if you separate a list into at its median, and then separate the list to its left by its median, and separate the list to its left by its median, and so on, you can make it very rapid to be able to look through this roster of numbers and see if a particular value were there. So you can perform a bisection search as well using Burrows-Wheeler to go hunting for a particular gene that hits a read. This is a whole lot faster than simply doing, let's talk about what my programming professor used to call a brute force and ignorance algorithm. So if we're doing a BFI approach, I'm going to take each read sequence. I have this read right now, and I'm going to see if it hits any of the genome annotation that I've got. Okay, so I can just take my one read and now search through the entire annotation for anything that's an exact match for it. Okay, you can imagine doing this. It takes a long time, but you can imagine how that would work. And then I could go to the second sequence and search that through the roster. Very, very slow though. So Burrows-Wheeler gives us a data structure that enables a bisection search of all the reads against all of the genome annotation, and it's much, much faster to do than any sort of brute force and ignorance approach. So Burrows-Wheeler has become very common in this application. So once we've done this, we now have assembled data. And we've already mentioned a file format just yesterday that would come out of this alignment process. Does everyone remember SAM and BAM? Okay, so everyone uses BAM files these days. So you can easily imagine then that having done a sequencing experiment, having run it through quality control, having trimmed off the junk sequences, having run an alignment, we now have a BAM file. This BAM file allows us to fuel things like the detection of variance. So I asked myself the question, how could we tell that a particular sequence base call is a real variant or not, based on the data we have? So in reading this paper, I came up with these sort of four basic ideas. First off, sequence coverage. I mentioned that we're going to hope for a whole stack of sequences at all of these locations that we hope to call variants. And this is where that vote is going to come into play. If you have just 2 reads covering a region, how comfortable do you feel saying that this G is different than the C that I expected to be there? Well, you're going to have to depend on some other points of evaluation, because having just 2 reads at that location isn't particularly good. Ideally, you'd have 50 reads that span this location, all of which agree that there's a difference here. So we want to see more reads to reduce our error of calling a variant. Next up, base calls declaring the change should have reliable Phred scores. Now, I think some of you, when I said, do you feel comfortable about the claim that this variant is present if you have only 2 reads there, were thinking in the backs of your minds, well, maybe they have great Phred scores. You're right. It is possible that these 2 sequences are in complete agreement that this is assuredly a G and not the C that the reference says should be there. So this is a second line of evidence that we can use, not just the number of sequences we've stacked up here, but by the fact that they're very, very confident about that letter. Thirdly, a sufficient number of reads. Okay, so this is another way of thinking about this. So maybe we have sequence coverage across this entire region, differentiated from the stack of letters on this particular location. So the reads may be all over this gene, but this count is just about this one column for this one base call. And of course, our friends in statistics are going to get involved here. So we need some sort of statistical test to evaluate the probability that the reference differs from what this person has at this location. That said, when we use modern sequencers to compare any person to the total genome reference for humans, you're going to find bazillions of differences for any individual human, simply because the human genome is so bloody big. So we really want to be very, very careful about miscalling sequence variants just because one read happens to show a different letter at some location. It's quite easy in such a huge volume of data for an individual letter to be wrong. Now, I spent a fair amount of time talking about high throughput sequencing here. Does that mean that we no longer use any technology but sequencing to detect variation? For this, I want to bring back the idea that microarrays actually have quite a lot of relevance. Microarrays have been used to power most of the very large N studies in Genome-Wide Association Studies. So if you're able to produce sequence variation data for all of the major SNPs that we know of in humanity, and can do this by microarrays, the volume of the data are quite a lot smaller. So I did a quick look at this last year to ask how many different companies are still in the business of microarrays. From the perspective of the bioinformatics community, you might be justified in thinking that microarrays aren't even sold anymore, but in fact they're doing quite a lot of good business now. The reason being that people sometimes just want to measure known variations across very large numbers of people, and that's a whole lot easier to do with a microarray. So what is a genome-wide association study? I've mentioned this enough times, I think. Let's imagine that we can amass 100 people who do have a disease that we think is associated with genes, and we've got another 100 people who do not have this disease. Is it possible that we can find genetic markers that are associated with disease risk? Now I note that I'm being a little fuzzy here. I've said markers, I haven't said the gene that causes this disease, right? So let's imagine that we have some gene that has a particular allele in the human population that is associated with this disease. We looked at a fine example of that this morning. The cystic fibrosis gene, cystic fibrosis is a disease that results from a particular allele that's typically missing the phenylalanine at position 508. So that allele, if you've got it on both chromosomes, is going to cause you any amount of awfulness in your life. So if we are looking for a genetic trait like that, Genome-Wide Association Association study is going to be ideal, because that's a monogenic trait. A change in one gene is all that's required to cause disease. And when you've got both of them knocked out with these poor alleles, you're going to have the disease. So it's highly penetrant as well. Most diseases are nowhere near that clear, though. So we need to be able to find markers associated with alleles that are deleterious for some disease. Now, I'm going to bring us back to a classic study in this space back in 2005. The guys who did this study and the women got a lot of stuff really, really right. And I think that this is one of the reasons why GWAS became so enormously popular in the last half of the first decade in this century. So in this case, they were interested in age-related macular degeneration. You don't want grandma to lose her vision, do you? I mean, it's terrible, right? So we would like to be able to solve diseases so grandma gets to keep her vision. So they have 96 cases and 50 controls. They were using microarrays for their measurements, looking at 116,000 different SNPs. 116,000. Now, today's microarrays for SNPs cover millions of SNPs. So this is a pretty minuscule subset of those by comparison. They found two associations were able to, which is to say two genes, or two regions of the genome associated with this disease, published in Science, and everyone said, this is how I want to do it. So I want to emphasize one of their plots in particular. This is called the Manhattan plot. Why is it called a Manhattan plot? People always fantasize about Manhattan that it's comprised entirely of skyscrapers. It's not true, but this is what people think. So imagine that all of these spikes represent towers pointing up into the sky. That's the Manhattan plot. So people in this case, we have a bunch of SNPs. Each SNP is some polymorphic single nucleotide change in the genome that is carried by, I believe, more than 5% of the population. Remember, we say that if a genetic variation is found in less than 1% of the population, we typically call it a mutation. Well, SNPs tend to be a little more popular than that. We don't give them the name SNP until a set number of people, a set fraction of people are observed to have it. So let us now sort all of our SNPs. We said in the prior slide that there were 116,204 SNPs that they were assaying. Now I want to note that some of these failed QC. Some of the SNPs failed QC. So they've thrown out a fair number of these markers as not being useful to them. But for the rest of these, they were able to compute p-values to say, is this differential or not, given the number of people, 96 cases and 50 controls. So again, we've already talked about a fine example where a low probability was expressed as a negative log. You can see that the people behind the Manhattan plot really like this idea. So here they're showing the negative log base 10 of the probability. So when something points up way high in the sky, that's to say this marker is one that we think has a significant association with the phenotype here, with macular degeneration. Grand. So when you see something spiking up into the sky, way above that, that's wonderful. However, there's this kind of interesting thing going on. We all see this dashed line across here, right? What is that dashed line? That dashed line is the threshold. Now, when you run yourself a t-test on something, what p-value threshold would you use? Typically. I'm asking, are men taller than women? I'm going to write down the height of every man and every woman in this class, and then I'm going to run a t-test on one versus the other. Some p-value pops out. What's going to constitute success on that t-test? What's that?
Being less than 0.05.
0.05. Okay, so this is what we teach in almost every case. T-test is giving us a difference between, is evaluating whether we would see means this different from each other given no real variation between them. If it says that the probability of that is less than 5% or 1 in 20, then we say, aha, our null hypothesis that men and women are no different in height is one that doesn't explain our data very well. So we overturn it. So, why are they doing all this nonsense? They're doing this because they realized we're not doing just one test in which all of these genes are compared. We're doing more than 100,000 tests across these. That is a lot messier. For SNP number 1, do we see a difference? For SNP number 2, do we see a difference? For SNP number 3, do we see a difference? On and on until you've beaten 100,000, you run 100,000 of these tests. Well, you would expect 5% of those to give you less than 0.05 p-values just by random chance alone. That's not very good. So we have to be much more careful, much more rigorous about what we're going to accept as significant. Here, they have set their p threshold at 0.05 divided by 103,611. They're doing that because they had 103,000 SNPs they were going to test. They've used something called Bonferroni correction, which is a very conservative way to protect you from making any mistakes at all in your claims about which SNPs are significantly different between cases and controls. So, having divided their p-value threshold by 103,000, they find that this line represents the probability of the probability threshold that these skyscrapers have to get over before we're going to call it a significant difference. So this was really nice. The Bonferroni approach meant that when they had these spikes up in the air, they were able to say, these are the ones we're going to claim, and anything less than that we're not going to claim. So Manhattan plots are kind of a standard output of Genome-Wide Association Studies, and they use the same negative log trick for visualization to emphasize those markers that have the most significant p-values. Now, how about this linkage idea? I mentioned a moment ago that the markers we were looking for, we hoped were going to give us some sort of clue as to which genomic features actually contributed to disease. That's not necessarily the case. These SNPs were not selected because they are part of genes. These SNPs were selected because they happen to be places where human genomes differ from each other. That does not mean they fall conveniently within some gene. When you do this analysis and you come up with these two, do you immediately get to a particular pair of genes that code for proteins with that SNP helpfully introducing a non-synonymous mutation in the middle of the coding sequence? Generally not. Generally not. Frequently we find that these markers are just hanging out in la-la land because after all, only about 1 in 100 locations within your genome fall within a protein coding gene. So we frequently find that these markers that we look for may be near a gene, or in the hinterland of a gene, or in a region marked as regulatory for some gene, but falling in a gene is actually perishingly rare when we're looking at SNPs. So I want to think about some genomic region as having a range of markers across it. So these RSs, think of those as SNPs here. Now I want to note that some of these SNPs are adjacent to others. They're pretty close to another one, maybe a few megabases off. And we see that there's a strong correlation in that people who have a marker at this location frequently have its neighbor as well. So this gives us the concept of linkage, genetic linkage. HaploView is a tool created by the Broad Institute that's a pretty useful way to visualize these data. It produced this diagram. So when you see something like this in a paper, I want you to think to yourself, anything that red triangle is hanging below, any of those markers rather, are pretty much interchangeable with the others. They correlate with each other because they don't get separated by recombination. Not very often. So genetic linkage is one of these things that allows for any of several markers to give us information that the next marker could just as easily have given us. When we pick which markers we're going to include in a microarray set, we're going to try to pick from a set like this just one representative, because it doesn't do you a lot of good to grab 2 different markers from the same linkage set. They're going to give you the same information back. We would like our markers that are chosen for microarrays to be just as informative as possible, thus including as many linkage groups as possible. Okay, that brings us to the GWAS catalog, and we spent some time there this morning. So the GWAS catalog is useful as a resource for understanding which studies have them already published in these for a given disease. Each of those dots is supposed to mark some disease that's been associated with some allele over time. It's an amazing resource, and you can really dig pretty far and find studies on most any disease that you find interesting. Now, I know we have a few genetics people in the room, and if I wander astray on some of these next few points, I hope you'll correct me, because I know that I want to make sure you guys are getting square information from me. So I've got several different concepts that I'm trying to convey here. One is linkage, the idea that two markers may be correlated with each other. The next one is stratification. Stratification is something that could challenge us. So I want you to imagine a scenario where I am working with my colleague in Beijing, Dr. Lee. Dr. Lee. Okay, so Dr. Lee and I are interested in the same disease. Is it asthma today? We'll study asthma? I'm going to study asthma. Dr. Lee and I are both interested in asthma. He has a cohort of patients who have some disease other than asthma, and another cohort of people who do have asthma. And I have people in my group of study who do have asthma and have some disease other than asthma. We want to go looking for genetic markers associated with asthma in our populations. And we think to ourselves, both of us are studying asthma. Let's throw our pools together, and then we'll have even more power for discerning these markers associated with asthma. Sounds good so far, right? Here's the problem, though. The people here in South Africa have different SNP collections, have different collections of SNPs present than do people in China. If you've got a strong Han Chinese population, and here I was working, for example, with the colored population, or with Zulus for that matter, it doesn't much matter. Any of these groups have very different kinds of profiles than what I'm going to find in the Han Chinese population. So let's imagine this scenario. I'm going to give you some numbers, all right, so you can write these down for me. So in the first case, I have an other group, and I have a asthma group. O and A. In my O group, let's say I'm working with Zulus. Do we have any Zulus in the room? No Zulus? Okay, that's fine. So I'm imagining I'm working with Zulus. I have 50 people who have some other disease, and I have 10 people who have asthma. All right, now my friend Dr. Lee is working with Han Chinese. He has 50 people working with other disease, and he has 30 people who have asthma. Did anyone notice a problem in those numbers I just gave? What we've done is to enrich our pool of asthma with Chinese, with folks from the Han Chinese population, and we've relatively enriched the other disease pool with Zulus. Now, when I look for a difference between asthma and other disease, some of the genetic variants associated with being Zulu look like they are genes instead for having some other disease, not asthma. Similarly, it looks like the genetic variants that are associated with asthma typically are those of Han Chinese people. So this is a problem. We've created what's called a stratified study. Stratification means that you've got the population blocks of your study have distinct ethnic groups within them where the genetic variation kind of runs in lockstep. There are certain genetic markers associated with being Han Chinese. There are certain genetic markers associated with being Zulu. And again, I'm just using these as examples out of the thin air. So this is really problematic when we run a statistical study of this sort, because now some of the genes associated with being Han Chinese look like they're associated with disease. So not okay. We have to be aware of stratification when you have characteristics within your population that within your study population other than the disease in consideration. Now, this gets even messier when you're working with populations that have been recently brought together over the course of evolutionary history. I know that things like the 16th century seem like a really, really long time ago, but from the point of view of evolution, that's an eye blink. An eye blink. So when you're working with something like the Cape-colored population here in the Cape, you'll see that that individuals have genomes that are kind of a patchwork then of the different populations that came together to create that created today's Cape-colored population. So you have some white alleles in there down from Europe. You have some Malay alleles in there. You have Khoisan alleles, certainly. These mixtures have created very similar kinds of statistical problems here when we're looking at the GWAS studies in the Cape-colored population, as the American genetics community has experienced with African American population. Because, again, we have a mixture of different continents' genetics coming together to create a combination of SNP markers that reflect both of the populations, or all of the populations that they came from. Read more carefully, you'd see that they have correlation among nearby SNPs. This reflects the regions of DNA that have been passed down intact. And segmental scale, so centenic SNPs, these are to say SNPs that fall in a given order on a given piece of DNA, have not been separated by recombination either. So both at fine scale and segmental scale, we find that the origins, the ethnicities that contributed to an individual are still perceivable in today's DNA for them. So how does that play out? Some of the research that's been done in this, that's been done in the Cape-colored population took place here in our own molecular biology and human genetics department. So when we look at, for example, Asian populations, European populations, African, here we're looking at the genetics of many people drawn from the Cape-colored population, and we're sorting them in different orders. So at first we're sorting them by the extent of Khoisan SNPs that they carry, the extent of the African, the Bantu-speaking cultures that have come down, the extent to which that shows up in their heredity, European contribution, and Asian population. So if you were to simply run a GWAS study on people drawn from the Cape-colored community, if you had failed to take into account what extent each person represents a contribution from the European population, a contribution from the Malay population, etc., your study's going nowhere. So you have to take admixture into account in order to do decent studies in these populations. Just one of the interesting ways, I think, in which history, a very complex and very, frankly, ugly history of what's been done to people in South Africa, how that very complex history plays into our attempts to understand how to help the health in these populations. Okay, so lots of closing thoughts here. The sequencing experiments are analyzed through a whole cascade of bioinformatics steps. Error propagates from each step. Now, I didn't say that in my slides earlier, error propagates from each step. What does that mean? It means that if I don't pay attention to my FASTQC reports, I might allow some sequencing experiments to move forward when I should have ruled them out because of their quality. If I fail to use Trimmomatic correctly to strip off sequences that are of very low quality, I may have compromised my alignment. Anytime you're using a very long, very complex series of tools to understand your data, you have a problem of cascading error. So it's best to make sure you get those first steps at least very, very right, because each of them can contribute error to your analysis downstream. Next, statistical considerations are inextricable from bioinformatics processing. Every bioinformatician, to some extent, must also be a little bit of a statistician. You need to understand these concepts. If you'd moved ahead in a Manhattan plot and said, Well, I looked at 0.05, but everything seemed past that. So I've now changed my threshold to use 0.0001 as my threshold. You would be in for a lot of hurt because the peer reviewers would eat you alive. So in using and in developing and in implementing bioinformatic tools, you'd better understand the statistics that allow their reports to be usable. Finally, I mentioned the high-throughput sequencer has eclipsed Sanger sequencing and microarrays, but both may yet be used to confirm its findings. Very frequently, we find that when we spot variance by massively parallel sequencing, we attempt to follow it up by using Sanger sequencing as well. And that is very useful for us in being able to confirm by an orthogonal sequencing technology whether or not there was truly a variant to be found at that location. So we have these options available to us. Certainly, if you're interested in doing a GWAS and want to collect dozens of people, sequencing them all may not be the best option, and simply for the data analysis piece. Working from microarrays might be a much more stable way to proceed. Okay, so tomorrow we're coming back to the fine world of genomics, this time thinking about what kinds of problems would be involved if we were sequencing a non-model organism. I think it'll be a lot of fun. It's a subject that I think gets us into some wild and woolly spaces in bioinformatics. We are back together again tomorrow morning at 9 for our practical in that area, and at 2 o'clock we'll have a quiz on everything that we've talked about today. So thank you very much. I'll see you tomorrow.